Thanks for the introduction and the opportunity to speak. And as we all know, cells are the basic unit of life. Now, how do they evolve in animals? So with single-cell atlases from almost every corner of the tree of life now, it has just become possible to approach this question. So the idea is to map cell types in organisms across long evolutionary distances. But the challenge here is that the complex evolutionary history of genes can blur cell type relationships. So let's first consider a conceptual scenario. An ancestral cell type is defined by its core-regulatory complex controlled by some transcription factors upstream of a series of target genes which form the functional machinery of the cell type. The cell type then diversifies into two daughter types, each now adding a new transcription factor into its core-regulatory complex. So this leads to the addition of new downstream functional genes and the replacement of existing parts. And as evolution continues, some genes might be lost, while others might expand to have duplicated paralogs. This causes parts of the gene regulatory network to be substituted by new paralogs, which further reduces the similarity between cell types. To solve this problem, so we have recently developed a cross-species cell atlas mapping method, which we refer as SEMAP, self-assembling manifolds mapping. So here's the idea. Consider species 1 and 2. So we begin with a gene-gene graph connecting homologs between the two species. Unlike other single-cell data integration algorithms that rely on presumed one-to-one gene orthology, SEMAP allows one-to-many homologs to account for gene duplication events. We then translate each species' features into those of its mapping partner to map the atlas manifolds, which we constructed by SEM. So SEM is a robust manifold reconstruction algorithm that we developed earlier. And based on the mapped manifolds, we then compute the spatial correlations of the gene expression for all homologous gene pairs and then reweight the homology graph using the correlations. So this cycle is repeated for a few times to gradually refine the gene homology and prune the many-to-many homologies to only account for ones that are expressed in the same mapped cell types. So to verify the method, we first apply SEMAP to the frog and zebrafish embryonic atlases collected in the Klein lab a few years ago. And these data sets have matched ontogeny, which can be used as a reference of comparison. So as shown here, SEMAP aligns the two atlases while maintaining high resolution for distinguishing cell types in each species, with a few example mapped cell types highlighted in circles. And SEMAP links correctly 26 out of 27 expected cell type pairs based on previous annotations, with the thickness of the edges quantifying transcriptomic similarity. And inversely, SEMAP does not link cells that are not developmentally related in most cases, suggesting a high degree of agreement between transcriptomic similarity and ontogeny relationships. A few notable exceptions, though, are secretory cell types that do share similar functions and common gene modules, which are highlighted by the black edges here. So these exceptions support the notion that cell types can be transcriptionally and evolutionarily related, despite having different development origins and even arising from different germ layers. I want to quickly mention that we have benchmarked SEMAP against several broadly used single-cell data integration methods and found that they only give minimal alignment between the expected cell type pairs. We also compared the results when restricting SEMAP to using only the one-to-one orthologs instead of the full homology graph. So even so, we identified similar, although weaker cell type mappings. So this suggests that at least for the frog and the fish comparison, SEMAP performance is owed in large part to its robust atlas stitching approach. So the key benefit of using the full homology graph is to enable the genome-wide identification of paralogs that have greater similarity in expression across species than their corresponding orthologs. So SEMAP identifies hundreds of such events with two examples shown here. So there's a minimal overlap between the orthologs pairs. In fact, in these examples, either zebrafish copy or the frog copy is only expressed. Strikingly, the missing orthologs are then replaced by one of their paralogs, for example, zebrafish SOX8A is substituted by SOX10, which has overlapping expression in neural crest cells with frog SOX8 with the co-expression highlighted in green here. And this substitution events can arise as the result of gene duplication followed by diversification of paralogs. In addition, genetic compensation by transcriptional adaptation, where loss of function mutations are balanced by upregulation of related genes, can also give rise to this type of signatures. And these results illustrate the potential in leveraging single-cell expression data to link molecular evolution to cell type evolution. So far, I have introduced SEMAP, which explicitly come for the complex evolutionary changes for gene families during cell type mapping. And in collaboration with many research labs, we have so far successfully mapped more than a dozen species. The mapping results appear to be highly consistent to experimental manual matching, and in many cases help the cell type annotation, especially in non-model organisms. But we have also observed, of course, limitations, especially when comparing species with extremely long evolutionary distances and completely different body plans. So these limitations may be attributed to the assumption that there should exist one-to-one correspondence, orthology of cell types between organisms. But this is only true if cell type diversification happens before speciation. In contrast, if cell type diversification happens after speciation, the diversification process should follow independent trajectories in two organisms. So in this case, one-to-one cell type correspondence may not exist, and instead we expect to observe families of related cell types sharing common gene expression programs that originated from the common ancestral state. So to test this, we mapped vertebrate embryonic data sets against two flatworms that my lab studies as a bilateral sister group of vertebrates. We also included two non-bilateral animals, hydra and a freshwater sponge, to sample a broader range of metazoan groups. So we performed all 21 pairwise mappings between the 7 species, connecting about 1,000 annotated cell types. So among all these mappings, we found neural and muscle-like cell types to show dense many-to-many connections among them, supporting the notion that ancestral cell types may have diversified into families over long evolutionary distances. To quantify the connectivity of the cell type network, we use transitivity, in which two mapping partners also link to at least one common cell type in a third species, forming triangles, with an example highlighted in red here. So more triangles does an edge belong to, better supported is that mapped cell type pair, and more densely connected is that cell type to its family. And indeed, neurons and muscles have high connectivity compared to the overall cell type graph, and all mapped pairs share a large number of genes, suggesting that the cell type families are defined by deeply conserved gene expression programs. And taking muscle cell type family as an example, so this includes not only muscle cells in all bilateral animals, but also sponge and hydra cell types that are non-conventional muscle cells, but have been implicated to drive animal contractions. So SEMAP identified broad transcriptomic similarities between all the cells, including genes from orthology groups spanning diverse functions, acting regulation, cell adhesion and stability, and signaling. I want to draw your attention to several transcriptional regulators shared among the cells. So previously, known core regulators involved in myocyte specification, such as MYOD, E12, and MAF2 are enriched only in bilateral and vertebrate muscle cells. Instead, we found homologs of muscle link protein, CSRP, and a FOX group 1 enriched in muscle cells from all seven species, indicating that the transcriptional regulation of muscle-specific gene expression may have evolved earlier than previously appreciated, and rely on a set of regulators that are different from what are typically assumed. So in particular, the majority of the muscle cell-specific FOX proteins are FOXC orthologs, which have been implicated to regulate cardiac muscle identity in vertebrates. In our analysis, they seem to be also conserved broadly among animals except the sponge. And notably, FOXC is substituted in subsets of muscle cells by FOXL and FOXG. And the FOXG is particularly interesting as it is enriched in 3 of the 4 invertebrates, suggesting that FOXG may play an underappreciated role in muscle cell specification outside vertebrates. So taken together, by mapping single-cell transcriptomic analysis, we identify hierarchical patterns in cell type diversification. We observe one-to-one cell type mapping between animals that are relatively close, typically in the same phylum, whereas among animals over long evolutionary distances, we observe interconnected cell types forming distinct families, which are defined by deeply conserved regulators. And I believe this is just the beginning, and a lot more discoveries are yet to come with more and more data sets becoming available to better sample the diversity of cell types in the tree. And this work is led by Alek Tershansky, a phenomenal graduate student, and our work has benefited hugely from collaborations with Dell Aron's lab at EMBL and Steve Quick's labs at Stanford. So with that, thank you for your attention, and I'm happy to answer your questions.
Thanks, Bo. That was very, very interesting and it's a really elegant method. I have a question about, under this framework you've developed, can you identify cases of concurrent evolution of cell types? What would you be looking for, and how would that be possible under this setting? So
for the concurrent evolution, you basically would expect sort of 2 cell types that sort of evolve in parallel. That's what you mean in this context.
Right.
Yeah.
Right. So getting to the same cell type with a cell type from 2 different lineages or 2 different evolutionary branches. Right.
That's right. So I think this is a great question. So, in this cases, you would like to compare cells that sort of in the embryonic development or in a sort of a constant homeostatic turnover that you would capture a lot of transient cell types. Now you could map the sort of terminal differential cells and the transient cells. And we do see parallel trajectories, like for example, when we compare the zebrafish and the frog data, because they have the cell types at different time points along the development, and we do see parallel developmental timing and the cell types in that case.
Thanks, Bo. Let's take one last question from Juan Lugo. Impressive work. Much of the studies are always considering large scales in evolution. What would be your predictions on smaller scales, like a group of sister species or even in phenotypically and genetically divergent populations of the same species?
That's a great question. So, as I mentioned, we observe 2 types of patterns in the cell type evolution. So within the animals that are relatively close to each other, we actually see a lot of homologies between cell types that we can draw one-to-one correspondence. And between animals that are very separate along the evolution, actually, you only observe cell type families which share ancestral gene modules. So what we really want to do is that we have data sets to cover both of these evolutionary scales, and with the animal that are close to each other, we can really construct the phylogenetic tree of cell types. While when you have animals that over long distances, you might sort of track how the cell diversity that are built in different branches of the tree of life. So one of the focus that our lab is focused on, because we work on flatworms, so we want to sample as many different flatworm species as possible so that we can reconstruct the trajectories of evolution of each cell types. I think the challenge here is that, when people are trying to decide which species to sequence to construct a cell atlas, they try to sort of distribute that effort as much as possible, right? Like they don't want to sample a branch of evolution repeatedly by sampling close related species. But I think this is going to come when we have a relatively good coverage of the tree of life, and then we're going to have data that concentrate on a specific branch of the tree.
Thank you very much, Bo Wang, for this excellent talk.